Hello, my name is Conway and I'll be presenting part one of this three-part presentation on the polymerase chain reaction. So what is PCR? PCR is an acronym for polymerase chain reaction and is a widely used lab technique for amplifying samples of DNA for further application and quantitative study. American biochemist Carrie Mullis invented the PCR process in 1983 and was awarded the Nobel Prize for Chemistry in 1993 for his achievements in PCR development and application in science. PCR is now so widely used in labs around the globe, it is considered to be the gold standard in synthesizing DNA for further study. The reaction is very sensitive and requires only a small strand of target DNA. Providing enough information is available to produce the primers for the process to run, PCR is also highly specific, and with precise primers, the chance of replicating the wrong section is extremely low. PCR works by copying natural processes that enzymes in our cells do every day. The first stage to a successful reaction is to ensure that you have a good sample of DNA. Now, provided enough information is available, the oligonucleotide primers can be synthesized. A separate process will produce the primers needed, and with the advances in genomics, scientists have access to many codon sequences to produce the primers that will be needed. Now, a note on primers used in the PCR process: DNA polymerase can only add nucleotides to pre-existing 3' OH hydroxyl groups. Primers are short nucleic acid sequences that provide the start point of the polymerase to synthesize DNA. Now, primers are usually 18 to 22 base pairs long, and by using both the forward and the reverse primers, the process is able to delineate a specific region of DNA to duplicate. Now, once a suitable gene or selection of DNA has been identified for amplification, what else is needed? Well, PCR is a very similar process to baking a cake. It requires a recipe of ingredients and a precise procedure to follow to ensure a good result. The main ingredients to note are the DNA template of interest, some DNA Taq polymerase, lots of oligonucleotide primers, that's the forward and the reverse ones, 3 nucleotides, these are the A, T, Gs and Cs, and finally a buffer solution containing cofactors that is balanced to around 8.4. All the ingredients for the reaction will be mixed and placed into small vials and loaded into a PCR machine called a thermal cycler. Now, before we go ahead and cook up some new DNA, we must first consider a few variables such as the type of polymerase used and some protocols that ensure incorrect or premature annealing does not take place. Modern PCR processes use a stable thermal polymerase that is procured from a bacteria found in thermal springs. Taq polymerase is found in the bacteria Thermus aquaticus and can handle the high temperatures that the thermic cycler subjects the DNA to during the cycles. Human polymerase takes a lot longer as operating temperatures would need to be lower. A Taq polymerase works fast and has a half-life of approximately 2 hours, which is more than enough time to amplify a DNA sample of interest. So since its inception in 1983, PCR has undergone several improvements to its fidelity to the point of being vastly superior to the original Mullis version. One process coined hot start PCR sees Taq polymerase inhibited until the first heating cycle. This stops the chance of premature annealing and extension from occurring. The benefit of this is the reaction can be set up at room temperature. Okay, so let's go ahead and make some DNA. So we now have the ingredients in the DNA sample of interest. What now? So the template DNA, some Taq polymerase, nucleotides and primers are all added to a buffer solution, and the mixture is then pipetted carefully into small vials and loaded into a thermic cycler. Now, it's important that labs follow strict policy and protocols to ensure that the solution does not become contaminated with other foreign DNA. Clean rooms are an absolute must when you're working with PCR. Now, PCR process is fairly easy and straightforward and is broken down into 3 stages. You have denaturing, annealing, and finally extension, or sometimes referred to as elongation. So stage 1 is denaturing. DNA needs to have its helical structure denatured, exposing the base pairs on the single-stranded DNA. The thermic cycler will heat the solution to 95 degrees Celsius for approximately 30 seconds. It is important to denature the double-stranded DNA completely so that the primers can work as intended. Denaturing breaks the hydrogen bonds between the 2 strands. If the sample has a lot of G and C pairs, the process will require more energy and time due to these bases having 3 hydrogen bonds between them. So now we're ready for stage 2. Stage 2 is annealing. During the annealing stage, the temperature of the solution is lowered to approximately 50 degrees Celsius for around 30 seconds again. The solution contains a huge amount of primers, and the cooling will allow the primers to anneal to the correct sections of the DNA. These sections are called promoter regions and serve as a start point for the Taq polymerase to do its job. Once the primers have annealed, the Taq polymerase will attach and begin the final stage of the reaction. Now, stage 3 is called extension, or sometimes referred to as elongation. During the final stage, the temperature is raised again to approximately 72 degrees Celsius to allow the polymerase to operate optimally. Taq polymerase will move in both the forward 3' and 5' direction, and also in the reverse 5' to 3' direction. Taq polymerase will add complementary nucleotides to the DNA section until a stop sequence is reached. The Taq polymerase, being an enzyme, is not used up in the reaction and is good to go again on another section of DNA. So, what did we end up with after all those stages? Well, after one run, each original sample produces 2 new molecules. Each new molecule will contain 1 parent strand and 1 daughter strand. If we were to use just one piece of DNA, a cycle of 30 runs would produce over 1 billion new DNA samples. The whole reaction takes anywhere from 30 to 90 minutes, depending on the machine used, the size of the DNA being replicated, and of course the number of samples that are going to be needed. So now the final product is ready for the scientific application that it was actually created for, and there are quite a few. So on that note, I would like to thank you for watching this presentation, and I'll now hand you over to Sarah, who will take you through the application of the newly created DNA. A quick note that all artwork in part 1 was created by author Conway Deacon, and full reference list will be presented at the end of the presentation. Thank you.
Thanks, Conway. Hi, I am Sarah Gray, and in this section of our presentation, I will be providing some specific examples of the applications and techniques of PCR and what happens after we have amplified the section of DNA that is of interest. After PCR has been completed, we have millions of DNA fragments at varying lengths, but what do we do with them? Where do we go from here? Well, today we will be looking at electrophoresis, and in particular, agarose gel electrophoresis. This is where an electrical charge is passed through a medium that contains DNA molecules, and they separate depending on their size and electrical current. Electrophoresis in the context of criminal investigations or DNA fingerprinting is what we will be exploring today. Let's have a quick recap of our DNA. Interestingly, 99.9% of our DNA can also be found in other humans. The remaining 0.1% might seem a minute amount of DNA, but it contains approximately 3 million base pairs that produce a pattern that is uniquely individual. This pattern is our DNA fingerprint. Let's use a recipe to make a DNA fingerprint via electrophoresis. Firstly, our ingredients. We need agarose gel, which is a polysaccharide polymer with a derivative from red seaweed called agar. It also contains repeated acarobia subunits, or sugars, and it comes in powdered flakes. We have DNA fragments, thousands of fragments in varying lengths. We have an electrical current, which is 1 to 5 volts per centimetre between electrodes, and constant wattage and voltage is also required. We need a buffer solution, which is a salt solution that will carry the electrical current. We also need dye and stain, ethidium bromide, which binds to the DNA, illuminates with a UV light, and banding pattern can be seen. We also have some apparatus listed. Let's have a look at our method. Okay, we have fingerprints from the scene of a crime, amplified through PCR. Now the process of electrophoresis can begin. Firstly, you may remember that our agarose gel comes in a powder. So the first step is to prepare our gel. So we fill a flask a third full with our buffer solution and add agarose powder using a weight to volume percentage to the solution. The concentration of agarose should be between 0.5% and 2% in the gel mix. We heat, melt and mix until agarose powder is totally dissolved. We add ethidium bromide at 0.5 micrograms per mil. We allow this to cool slightly, and pour into gel casting tray and place a comb into the gel to create wells for the DNA. The comb is removed when gel becomes solid. The gel is in the form of a permeable matrix through which DNA molecules can travel when an electrical current is added. Keep in mind that DNA may be different sizes, so viscosities of gel may be altered to suit the DNA and change the size of the matrix. Many scientists now use pre-made gels, so they're not required to complete this step. Next, we add a loading dye to the DNA, namely 30% glycerol, 0.25% xylene canol, and 0.25% bromphenol blue. This will track the DNA sample as it travels and will add to the viscosity of the DNA. The agarose gel is now placed in the electrophoresis tank, and the buffer that was used to mix the gel is also added to the tank until the gel is submerged by the buffer solution. A DNA marker or ladder is placed in the first well and is used as a baseline measurement for the unknown DNA fragments. The DNA samples are then added to the remaining wells. The electrophoresis tank is covered. Power supply and electrodes are attached, negative on the side of the sample wells, as we can see in this diagram, and positive on the opposite side, so that the negatively charged DNA travels through the gel to the positive electrons. We let this run for an adequate amount of time, approximately 1 to 1.5 hours, and then we can compare the bands to the marker DNA and size up the base pairs. Shorter pieces of DNA will travel through the gel matrix faster than larger ones. Longest fragments of DNA will be at the well end, and the shortest fragments will be close to the positive end. Now separated, fragments under UV light will glow, and bands appear at different points along the length of the gel. Fragments of the same size have migrated together in groups to band together in the same position. The distance the DNA has migrated in the gel can be judged visually by the migration of the buffer dye. Base pairs can be measured by our DNA markers while they are still in the gel. However, we have one more step. DNA is finally transferred from the gel to a nitrocellulose paper and then to short radioactive pieces of DNA called probes, which are short sequences of repetitive DNA between 10 to 60 base pairs in length. These probes only bind to sections of DNA that they are complementary to, and when the nitrocellulose paper is X-rayed, the regions of DNA that bound to the probe will appear, and that will reveal our DNA fingerprints. And we have a match. Here we have an illustration of a crime scene, and our crime scene prints match our suspect 2 prints. This is just one of the many techniques that PCR is responsible for making faster and more accurate. But there are many, many applications to PCR. We have genomic cloning, where PCR is used as an alternative and adjunct to standard cloning procedures. We have genetic engineering, where PCR has simplified engineering techniques. Prenatal diagnosis, we're using PCR on amniotic fluid to diagnose congenital toxoplasmosis. Molecular archaeology, where PCR has been used to extract ancient DNA from a wide range of materials. This concludes my presentation on the application of PCR, but we are not finished yet. I would like to hand over to William, who will continue our presentation through its third and final section. Thank you for listening.
Hello, my name is William, and welcome to part 3 of the presentation, where I will be talking about the advantages and limitations of PCR in general, as well as the future for implementation of PCR techniques. PCR, as with any procedural technique, has its advantages and limitations. These should be retrospectively considered when analyzing what procedure is best used in their respective situations. Over the following slides, we will be investigating each advantage and limitation listed. PCR is a highly sensitive technique, known to be perceptive of any change made to its target. Only the smallest amount of DNA is required in order to fully amplify the sample, resulting in the production of millions and even billions of replications of the target DNA. Additionally, Due to the minute amount of sample DNA required, PCR allows for greater flexibility with analyzing old, potentially degraded samples of DNA, allowing for scenarios such as post-mortem tissue analysis, particularly useful in crime scene investigations. PCR is well known for its simplicity, especially in comparison to other techniques that serve the same purpose for DNA replication. It is also relatively cost-effective and is not time-consuming. A notable example would be in detecting Leishmania tropica, a known viral parasite in humans, via high-resolution melting PCR. PCR produces results rapidly. This is most in part due to the way PCR replicates DNA, allowing for rapid replication. This is especially helpful when results are time-sensitive and vulnerable if otherwise left to their own devices. This also means that experiments with PCR implementation can be done in shorter time, allowing for multiple renditions in the allowed time. PCR, however, for all its advantages, does have notable limitations. For one, it requires prior information of the sequence in question. As the experimenter must create appropriate primers for each end of the DNA sequence, they must have prior knowledge on the sequencing at the region specific to the experiment. DNA polymerases are prone to error, which can inadvertently result in unwanted mutations. Experiments such as cloning of microorganisms or even other animals require highly accurate amplification of DNA for success, otherwise it may lead to undesirable outcomes. However, errors can be mitigated using high-fidelity DNA polymerase that is appropriate for the experiment in question. As noted earlier, PCR is a sensitive technique. However, this in turn presents a double-edged sword scenario. Weisenegger explains that even in the smallest contamination can result in a misleading outcome that can violate the reliability of any recorded data specific to the experiment. Such an example will be seen in cases where agrobacteria that are present in plants are accidentally amplified instead of relevant plant DNA. Regardless, the future remains bright for PCR as new avenues for its usage continue to present itself in a variety of ways. Qualitative detection of rare fetal-specific events is a potential future application of PCR, specifically droplet digital PCR. ddPCR, as opposed to qPCR or real-time quantitative PCR, would be the preferred method due to its high sensitivity to the develop adapted assay, making it a lucrative qualitative alternative as opposed to qPCR, which is known for its highly variable results and work-intensive demand for reproducible outcomes. A 2015 study presented by Silence and Co. showcased through ddPCR usage a non-invasive method that could provide a highly accurate reading into both RHD genotyping and fetal sex determination. This information can thereby be used to identify potential maternally linked disorders, such as Duchenne muscular dystrophy, as well as limiting therapy implementation towards male fetuses intended for treating females with congenital adrenal hyperplasia. Avoidance of invasive methods can overall reduce risk to both fetus and the mother, which makes the alternative potential highly warranted. ddPCR utilization can also make cost-effective detection of fetal aneuploidy a reality. Discovery of abnormal numbers of chromosomes is currently a great costly process, with next-generation sequencing the only method available at this time that yields satisfactory outcomes. Differential methylated loci investigation is another potential future application of PCR. TIMP3 promoter region that is hypomethylated in DNA from pre-eclampsia placenta relative to DNA from maternal blood has reportedly been seen as a potential diagnosis marker for detection of pre-eclampsia and maternal plasma. By using digital PCR, it is possible to quantify differentially methylated loci after pre-analytical step of methylation-sensitive digestion or biosulfite treatment. This serves as an effective method for diagnosis and monitoring pregnancy-associated disorders like pre-eclampsia. Thank you all for watching, and we hope you enjoyed our presentation on PCR today.